Okay. So welcome everyone to the last video of this week, module 3.3 demonstration. And in this demonstration, we'll do the multiple sequence alignment using Clustal Omega. Now for that, what we will require, okay, we will require some sequences, okay, and that sequences I have gathered here. You can see here on my screen. Here I have gathered all the sequences from the hexokinase. These all are the hexokinase, that is the enzyme protein which works as the first enzyme for the glycolysis hexokinase. I hope you know about it. And I have taken all possible sequences. Suppose here I have taken Drosophila melanogaster, Diplocarpon rosae, Coletotrichum chlorophyte, okay, Epispora, then there are Abridopsis thaliana, Streptomyces polyasparticus, then Klebsiella pneumoniae, Rhizotonia solani, Oryza sativa. So many different and non-related species I have taken. So this kind of thing. Just 1 minute. I will share this with you. Okay. Now what we'll do, we'll go to the Google Chrome and we'll see about how to do the or how to work on the Clustal Omega. Now here I will search Clustal Omega. Okay. And here I will open the first ever site. Now here I have saved this file, that file you just saw. Due to some mistake it goes blank, but I have saved this file as a MSA input file text. Okay, I have saved it in the text format. Otherwise, what you can do simply, you can simply copy the entire file, entire sequence, and you can paste over here. I have created it in the text format, so I will use that file directly. Okay. Now here I will choose the file. In the desktop I have this file, so MSA input file. See here, you can see the name, MSA input file, and here I have to choose the Clustal Omega with character counts. Okay, that we will choose output format. Um, Other options are there, but at this moment I will not suggest you what to do. Okay, okay, I want distance matrix, so I will do this. But yes, at this moment don't do anything, just keep as it is, just use the whatever is already there, and I will give one name, hexokinase enzyme MSA. Okay, and we'll submit. Here you have to uh, change. Okay, just 1 minute. Okay, here you have to check protein, DNA or RNA. Okay, so we are working on protein. Hexokinase is the enzyme, it's a protein, so I have checked the protein. If you are working with DNA, check DNA. If you are working with RNA, check the RNA. Then I will submit it, so it is getting ready. This job ID is important. If your job is taking so much time, just copy paste the job ID. Okay, now here you can see the thing. Okay, and here you can see the output. Okay, now I want the color output. Just 1 minute. Here you can see colored sequences show, And you can see very, if you see the very closely, now look at this sequence. Okay, look at this particular, this thing. Okay, this here, can you, if you are seeing my cursor, look at this particular region and this particular region. Also look at this particular region. You can see, in the many species the sequence is same, the position and sequence is same. That means these things, these regions are the conserved domains. Okay, these regions where the you can see the sequences are common, this is called as the conserved domains. Okay, and this shows these positions are common. Now we'll go to the alignments. Here you can see more or what we can say, more visual. Now I'm just minimizing the things. Now you can see just by the coloration, you can see here. Okay, or I will choose the Clustal 2 here only, so it has the color combination very significant. Now see, look at this table. Okay, in this region and this region. Okay, also we'll move little bit forward. Okay, this region. Okay, these regions you can see, also see this one. Okay, this one. So these regions are common. Okay, the sequence at these regions where you can see the same color line is common throughout many species. So this much species we have taken, so throughout this species the sequence is common. That means the these regions are conserved domains. These domains are conserved, these chunks of that protein or amino acid sequence is conserved. Now coming to the guide tree. Now see here, very important thing over here. Guide tree means it shows how the algorithm worked to find out this analysis, to find out these outputs, these alignments, etc. Okay, how this entire algorithm worked. So first of all, it took the Klebsiella, then it took the Drosophila, Arabidopsis, Oryza, Streptomyces, then Colectricum, then Epispora, like that. So this particular phylogram shows us that how this algorithm have worked. Okay, actually we have to deal with phylogenetic tree. Now what I will do, I will just little bit increase the size and the, I will maximize the thing here. Now you can see here, you can see the species name and one number is there. I will tell you. Now look at this position. Okay, look at this position. Just 1 minute. Okay. Now see, this position shows there is something who is the common ancestor of everything. Okay, who is the common ancestor of everyone. Now here coming to the Klebsiella and this junction shows there is someone who has the common ancestor between these two. It may be algae, right? It may be unicellular algae or something. Arabidopsis and Oryza, these are the plants. So we can say that, or this thing is about the hexokinase. So there is some organism which shows the similarity, or which serves as a common ancestor for this enzyme. Okay, it doesn't mean that totally it will have the common ancestor. No. Then Arabidopsis. Now here you can see that for these two groups, this group and this group, there may be some common ancestor for these two. There may be some common ancestor for this group and this streptomyces. There may be some common ancestor, and for this and this there may be some common ancestor. Now, how to calculate the changes in the enzyme? Now, suppose I want to calculate the change between Arabidopsis and Oryza. What I have to do? I have to find the common ancestor between them. Who is the common ancestor between them? So, in Arabidopsis and Oryza, you can see this is the common ancestor. Now, what I will do? I will calculate 0.28 and 0.23. Okay, if I calculate it, 0.23 and 0.28, it about 0.51. Okay, it about 0.51. So it shows 0.51 substitutions per amino acids or per sequence. So, or in the some simple words, what we will do, we'll just multiply this 0.51 into 100. Okay, so we'll get the 51%. That means this Arabidopsis and Oryza have the changes in their protein sequence by the 51%. So, or what we can say, 51 substitutions per site. So that kind of thing is there. Now here Klebsiella and Arabidopsis. If I want to calculate Klebsiella, 0.49 plus 0.06, so it becomes 0.55, and this 0.28, so it becomes 0.80 something approximately. So about 80% proteins are different from Klebsiella and Arabidopsis. So that's how you have to calculate. If you are not getting this thing, just revise the video one more time. Then results viewer, if you want to change this, so you can do this. Then results file, if you want to download these files, you can download it. Then submission details, you can see the what are the submissions and everything. So that's it all about the multiple sequence alignment. If you have any doubt, you can reach me, and by the Google form that I have shared with you. And that's it from my side. If you have any doubt,